Alright, so welcome back to EBE. Last time what we did was talk about the most common type of photosynthesis called C3 photosynthesis. Now it's called C3 photosynthesis because the first detectable product of carbon fixation was two 3-carbon PGA molecules. Now, we did have a 6-carbon molecule produced because we were combining 1 carbon from CO2 and 5 carbons from RUBP, but because this isn't detectable, or wasn't detectable, because it's so short-lived, it was called C3 photosynthesis because the first detectable product was PGA, 2 molecules of PGA. Okay, but we also discussed last time how in this C3 photosynthesis, the enzyme that's used to do carbon fixation, Rubisco, has a dark side, and that dark side is that it's capable of fixing oxygen just as well as it is capable of fixing CO2. If CO2 is fixed, photosynthesis occurs, or carbon fixation occurs. On the other hand, if oxygen is fixed by Rubisco, the plant is going to lose a carbon and an ATP in trying to get its carbon back where it started. So I kind of mentioned last time, well, if we have this system that's not operating very well for the majority of plants, we've had a long time to try to solve the problem, right? Plants arose about 450 million years ago. You would think in 450 million years, plants would have solved this little problem. But my comment to you was, this is the enzyme used not just by all plants, but all photosynthetic organisms, including cyanobacteria, prokaryotic photosynthesizers, and that this mechanism seems to be very fixed, and maybe it isn't possible to alter the affinity of Rubisco for CO2 without also altering its affinity for oxygen. That is, maybe there's no way to solve this problem by fixing Rubisco. So, today what we're going to talk about is a couple other types of photosynthesis that exist out there where plants have solved this problem, but they haven't solved the problem by solving the Rubisco problem. Instead, what they've done is come up with a second carbon fixation reaction. So, let's take a look at that main reaction. This reaction occurs in a group of plants called the C4 plants. C4 plants are going to be doing what we'll call C4 photosynthesis. What we'll see is the C4 photosynthesis is just a preceding reaction before the C3 reaction. It's not as if they're doing a whole different type of photosynthesis. Instead, what these plants are doing is an extra bit. This reaction is the one that's unique to these C4 plants. The C4 plants start with a molecule called PEP, phosphoenol pyruvate. You may remember PEP from glycolysis, maybe not, but it is one of the molecules that occurs in glycolysis, and in fact, if you convert PEP to pyruvate, you're talking about the last step in glycolysis. So there's this PEP molecule that's hanging around in plants, probably because it's produced in glycolysis. And what we're going to do is, using an enzyme called PEP carboxylase, we're going to take and fix a carbon onto PEP. Now PEP has 1, 2, 3 carbons. CO2 has 1 carbon, and that means that the molecule that we're going to manufacture through this carbon fixation reaction is in fact a 4-carbon molecule called oxaloacetate. Now, for us, this oxaloacetate aspartate line, I'm not really interested in that. Instead, what I'm interested in is this stuff over here, which is malate. So I'm going to rewrite the reaction like this to accentuate this idea that it's in fact malate that we're after. Okay? All right. So let's take a look at these C4 plants and incorporate this reaction into these plants. So this is a leaf of a C4 plant. All of the C4s, well, not all of them, the majority of them are grasses. There are C3 grasses, there are also C4 grasses. So in these C4 grasses, they have this weird anatomy where around the xylem and phloem within the leaf, there are these cells called bundle sheath cells. And these bundle sheath cells are unique to plants that do C4 photosynthesis. Okay? Now let's take a look at this in a larger picture here. We're again still inside this leaf. There are cells within the leaf called the mesophyll cells, and there are other cells that are bundle sheath cells, etc., etc. But let's just take a look at this reaction. So we're going to start out here with PEP, right there. There's the CO2 coming in and the production of oxaloacetate, and then what I said, malate. That's what we're really interested in. Now, this is done out here in the mesophyll cells, okay? In the mesophyll cells, we do this carbon fixation reaction to make malate. Now what we're going to do is ship the malate over here to this bundle sheath cell. So in our diagram here, there's a mesophyll cell, there's another one, and we're going to make the malate here through carbon fixation, and then we're going to ship the malate to the bundle sheath cells. Okay? Ship the malate to the bundle sheath cells. Now, the malate is going to be converted to pyruvate. Remember pyruvate from glycolysis, same molecule. So this malate has 4 carbons. We're going to switch it over to pyruvate, which has 3 carbons, and in doing so, kick off a CO2. That CO2 is produced in the bundle sheath cells. This CO2 is picked up by RUBP in the presence of rubisco to make PGA and ultimately PGAL, and that's where we're doing the production of these organic molecules. Okay? The pyruvate is then shipped back to the mesophyll cell here, and the pyruvate is converted to PEP by using ATP to do it. Okay? So it's like we have this little racetrack going on. All right? Over here in the bundle sheath cells, right? And over here, sorry, this is mesophyll. Over here in the mesophyll cells, and over here in the bundle sheath cells. PEP is converted to malate, is converted to pyruvate, is converted to PEP, is converted to malate, is converted to pyruvate to PEP. So what we have here is a little carbon shell. The PEP, in being converted to malate, we move the malate and move the carbon with it, crack the carbon off, go pick up another carbon, drop it off, pick up another carbon, drop it off. Okay? So it's almost as if what we're doing is this carbon fixation reaction in one part of the plant and this carbon fixation reaction in a second part of the plant. This carbon fixation reaction over here is the one that we've done before, the RUBP to PGAL in the presence of rubisco. But in doing the reactions this way, because PEP carboxylase only grabs carbon, it can still do this reaction in the presence of lots and lots of oxygen. We don't have to worry about photorespiration. So they pick up the carbon, and then what we'll do down here in the bundle sheath cell is to try to keep the amount of oxygen low in this portion of the plant. And that way, the carbon that is cracked off, we can crank up to higher concentration relative to the oxygen and improve the likelihood that the rubisco picks up the carbon. Okay? And one of the ways we're going to do that is by limiting the amount of oxygen that's produced here. If you look, you can see all this thylakoid membrane in these chloroplasts. And up here, look at all those grana. This part of the plant does lots and lots of light reactions. Oxygen is a waste product of those light reactions. But we're not doing them down here, so we're not manufacturing oxygen in this portion of the plant. We're trying to keep the CO2 high to the oxygen to improve the likelihood that we fix the proper one. Now, it turns out this is not an ideal solution for all plants. Otherwise, this little trick probably would have spread around the planet because these plants would have a higher fitness relative to the C3s. But the problem is this little step right here. It actually costs energy to do C4 photosynthesis, whereas the C3 photosynthesis does not have this same ATP requirement. Okay? The rest of it, this portion, is the same for both C3 and C4s, but this little extra step here, plus the shuttling of all these carbon carriers around, increases the cost. So it turns out that this type of photosynthesis is really only common in parts of the world that are fairly arid. So the C4 grasses occur in places where there isn't a lot of rain, grasslands that don't get a lot of rain, whereas the C3 grasses tend to occur in places where they get more rain. Okay? Right, so what we've done here is we've spatially separated first carbon fixation using PEP carboxylase from the second carbon fixation that uses rubisco. Okay? Right, that's the first group of plants that's C4 photosynthesis. The next group of plants that has found a solution to this problem are the cacti and succulents. These are species that belong to this family called the Crassulaceae. This includes cacti and succulents. Succulents are also arid plants. They have very rounded leaves. They're not flat, high surface area. They're low surface area to volume leaves, and they're full of water. That's why they're called succulent. Okay? Right. They do this type of photosynthesis called crassulacean acid metabolism, or CAM photosynthesis. Okay? CAM photosynthesis, in my mind, is exactly like the C4 one. It's just, instead of separating the two carbon fixation events into different parts of the leaf, we're going to do the two carbon fixation events at different points in time. So, at night, the cacti open up their stomata. During the day, they keep them closed. And that may make some sense, right? The hotter it is outside, the more water you would lose if you opened up the stomata. So the cacti open them at night, where they tend to lose less water that way than they would if they did them during the day. So you open up your stomata and CO2 can diffuse in. This is at night. And what you do is you do your PEP to malate reaction. And this is done during the night. Sorry, just checking my, getting toward the end of these markers, I think. So PEP, in the presence of PEP carboxylase, picks up a CO2 to make, ultimately, malate. Again, I skipped the oxaloacetate step. We're making malate. The malate is then stored in the central vacuole through the rest of the night. Then, during the day, stomata are closed, no more CO2 is going to enter from the outside world. No more oxygen is going to enter from the outside world. Then what we're going to do is take out that malate, and we're going to take off the carbon. And when you take the carbon off of malate, you get pyruvate. The pyruvate then gets converted back to PEP. Okay? All right, now the carbon that's been released as CO2 from the malate then enters into the Calvin cycle during the day when the stomata are closed. And if the plant cracks off a bunch of carbon off of these malates, it can increase the concentration of CO2 inside the plant to a higher ratio of CO2 to oxygen than would normally be possible, given that there's a lot more oxygen in the atmosphere than CO2. Okay? So do this reaction during the day, this reaction at night, and then during the day, release the carbon and do the carbon fixation reactions that we talked about before as part of photosynthesis. Okay? All right, last thing to communicate with you. These two groups of plants, the C4 grasses and the Crassulaceae, right, the cacti and succulents, have figured out a way to do a photosynthesis that saves them losing a lot of ATP and carbon that they've previously fixed. But because of the additional ATP costs that we incur in converting pyruvate to PEP, the cost can be greater than the benefit under some circumstances. If the plant has abundant water, it appears that this cost outweighs the benefit. But in dry regions like deserts and prairies and grasslands that don't get a lot of water, C4 and CAM photosynthesis, the benefit outweighs this additional cost. Okay? All right, now, the last 15, 20 years, researchers have been trying to figure out the genetics of these types of photosynthesis. If I